So today we're going to have a look at PretextView. So we go to load map and then we can navigate to our file. And this is the map we want to load. So we press U and that gets rid of the menu. Okay, so this is our entire map, zoomed right out. So what we can see is we've got lots of scaffolds. The scaffolds are divided up by a grid line. Because we've got the tooltip switched on in our menu, as we hover over each scaffold, it will tell you the scaffold name and the coordinates that we're in at. Most of the scaffolds appear to be entire chromosomes. We can tell that. For example, if we look at scaffold 4, this one, if we zoom out and look across the whole map, there's no association between scaffold 4 and any of the other scaffolds. So that tells us that scaffold 4 is in fact an entire chromosome. And looking at the high C map of scaffold 4, it looks to be very well assembled. What we're looking for is that all of the strong association indicated by this red signal is on the diagonal, and there's no red signal off diagonal. And that's what we see. So all of scaffold 4 associates with itself broadly, and it doesn't associate with, for example, scaffold 3. And the strongest association is along the diagonal, as we would want. So this isn't true with all of the scaffolds. So you can see if we have a look along scaffold 1, we can see association between scaffold 1 and scaffold 33. And when we zoom in to have a look at the strongest association, we can actually see 2 points of very strong association. And these, so if we look at the top of this one, it's around coordinate 40.92 megabases in scaffold 1. If we have a look at the bottom of this one, it's around the same coordinate. And this leads us to suspect that there might be a gap in scaffold 1 at about 40.92, and that scaffold 33 might fit into that gap. So we can test that hypothesis. So we can press E, and we middle click, and that will select the whole scaffold. So as we zoom out, we'll see it in a minute. There it is. And it's moving towards scaffold 1, where it wants to sit. And we can see where the strong signal wants to take us is a break in the signal in scaffold 1. And so if we put scaffold 33 into scaffold 1 at this position, it looks really good. So if we left click, we'll drop it into scaffold 1, and then we can press E and zoom out and have a look at it. Yeah, that looks nice. So there's strong association between the left end of scaffold 33 and the point that we've placed it into scaffold 1, and likewise between the right end of scaffold 33 and where we've placed it in scaffold 1. There is some off-diagonal association here, but that's between what I would assume to be a repeat around this point in scaffold 1 and the sequence at the other end of scaffold 33, which is likely why scaffold 33 was left out in the first place, because there's a repeat that the scaffold has struggled to correctly resolve. Okay, let's have a look around our map, see what else we can find. Okay, so there's another strong one here, and it looks to be the same thing. So again, we've got a strong signal at one end, strong signal at the other end, to, trying to associate to one particular place in scaffold 2. So we do the same, press E, middle click, and move the little scaffold into scaffold 2. And again, it wants to drop in where there's weak association in scaffold 2. And we can see where it wants to go. So in reality, when we're curating like this, we wouldn't just be putting the pieces in like we are doing here. We'd also be editing a TPF. So TPF stands for tile path format, and it allows us to actually edit FASTA by moving chunks of the assembly around. And what we'd expect to see is where we want to locate scaffold 42 into scaffold 2, at the coordinate that we've broken scaffold 2 to put scaffold 42 in, we'd expect to see a gap in our TPF. And that would be really good evidence in conjunction with what we're seeing in the high C map to place scaffold 42 precisely at this location. Not only can we place it like we have done there, if we deemed it necessary, we could actually complement our scaffold. So I've just picked it back up again by pressing E and middle clicking. If we hit the space bar, we can complement it. Now, clearly that looks wrong. You can see the right-hand end of scaffold 42 wants to associate with the right-hand end of scaffold 2, not where we've got it at the moment. So we want to put it back how it was. But I was just showing you how you can complement something. So we press E, middle click, and flip it. Okay, so that looks really good. So we're just going to keep going around our map and finding things to improve. So we've got some very strong signal here with this small piece and scaffold 3. So I'm going to press S, and what that does, it puts me into snap mode. And snap mode means that I can't move the piece into a scaffold. I can only place it between scaffolds. Now, this piece clearly belongs here. It associates with the left-hand end of scaffold 3, but it's in the wrong orientation. We can see that because the right-hand end of scaffold 53 has stronger affinity with the right-hand end of scaffold 3 than the left-hand end of scaffold 53. So we can hit the spacebar, and that flips it, and you can see that looks better. So coming out, there was another scaffold that wanted to go with scaffold 53. Here it is. Pick that up and move it. Again, we're still in snap mode because we haven't turned it off. That's what we want to be in for this. So it could go that way around, but it looks much better that way around. So that's what we're going to do. Okay, so we've got another example here. We're going to move that and we're going to drop it into scaffold 5. Now we want to come out of snap mode because we want to go inside scaffold 5. So hit S, and you can see there's a break in association where it wants to go. But you can also see it's in the wrong orientation at the moment. If we hit the spacebar, you can see it looks right now. In that orientation, it's clearly wrong. In that orientation, it looks right. Again, we'd be examining our TPF file to make sure that there is a place for scaffold 45 to insert into scaffold 5, and we'd expect to see a gap at about 23.58 megabases in the TPF file. So going down, there's another one, more of the same. So we're just going to drop this in where it belongs. Again, looking in our TPF at the same time, making sure that there's a home for this. This one has strong association between the left-hand end of scaffold 48 and the right-hand end of 16. So this won't need to be complemented, and we want to place this in snap mode. So we hit S and just drop it where it is, and you can see that looks good. So we keep doing this. Interesting. Okay, so what have we got here? So the whole, because we've used multi-mapping reads to generate our pretext map, any repeated sequences will get reads mapped to both copies. So I'll show you what that means. So let's just move this piece where it belongs. So we're still in snap mode. So it wants to go at the left-hand end of scaffold 17. I'm just going to flip it so that it's oriented in the same direction. So to me, this looks like haplotypic duplication. So the reason I say that is that the whole of scaffold 54 appears to be duplicated at the beginning of scaffold 18. And this is where we want to examine our coverage track. Okay, so if you hit U and go to extensions and turn coverage on, and we'll just zoom out a little bit. And so you can see normal coverage is at this level, but the region that looks to be duplicated is consistently at a much lower level. So we're getting coverage in the 20s as opposed to coverage in the 30s, maybe it's less than 20s, yeah, maybe high teens and maybe 30s, something like that. So let's turn the coverage back off again. So it seems fairly clear cut that the coverage in this whole region is roughly half what it should be. If this scaffold were taken away to the haplotype bin, then if we were to remap our reads, we would get normal coverage across this whole chromosome. So this is haplotypic duplication, which we can remove. So for now, I'm going to leave that there. But in my TPF, I would be taking scaffold 54 out, and I'd be putting it in a different TPF assigned to haplotypes. Okay, so I'm going to keep tidying the map up. Okay, so there's a little bit of uncertainty over the structure of this particular chromosome. So at this point, I'd be looking for additional pieces of evidence. So something you could look for would be telomeres. So if I've got the structure of this chromosome correct, it would be reasonable to expect to find telomeres at the left-hand end of scaffold 39 and the left-hand end, because this is in the reverse orientation, of scaffold 32. So we know that scaffold 32 is in the reverse orientation because the label line here is on the bottom. When the line is on the top, it means it's still in the plus orientation. When we flip it, this line comes to the bottom. So any uncertainties as we're rearranging our map, we should make a note of for further investigation. I'm fairly happy that this is assembled correctly, but I'd want to be looking for additional evidence that I've got the join between scaffold 28 and scaffold 51 correct, and that the telomeres are here and here as I've got them. Looks like we might have a little bit of haplotypic duplication between the end of scaffold 30 and the end of scaffold 29. You can see there's just two direct repeats apparently here. Again, we would just turn our coverage on and just check that this is a bit low. So let's do that. And indeed it is. There's a dip in coverage just there. So we'd want to be having a detailed look at that and trimming the end of one of these two scaffolds to take some haplotype away to the haplotype TPF. So again, we've got some more retained haplotypic duplication here. So there's a region of mutual exclusivity between this point down to here and this point down to here. So again, let's turn our coverage on. And we can see that the coverage drops to about half, the same as it has done where we've identified haplotypic duplication in other parts of the map. So again, the correct thing to do here would be to perhaps remove this whole scaffold to the haplotypic TPF and also trim the end of scaffold 30. Once we've done all that, our map is pretty much done. We just need to see if we can account for these few scaffolds here. So we can do that by going up the y-axis and see if there's anything that we've missed when we were doing the broad scale stuff. So here's one. This needs to go into scaffold 15. Take snap mode off. And again, we'll be looking in the TPF for exactly where this goes. So there should be a gap here, and we should check that. What we should do as well, because our resolution isn't very good here, so it's not entirely clear which orientation this scaffold belongs in. So we could have a look in high glass at the association between scaffold 15 and 56. The higher resolution in high glass would likely enable us to get a definitive orientation for scaffold 56. So that would be something that we'd want to check in high glass. So continuing to look up the y-axis, and we can see a very clear association between 57 and 11. There's a big hole in the association here, suggestive that this is actually haplotype. So let's just double check that. Snap mode on, yes, and we'll drop it into place. And so what I'd be expecting to see if I were to look at in the telomere track here is we'd probably find some telomere on the end of this and on the end of this. If we were to do, for example, a dot plot of this scaffold against this scaffold, I'd expect most of this to be contained within this scaffold. Again, I'd expect our coverage to show us that this is lower than you'd expect. And again, it is. There's a dip in coverage here. So I'd be pretty confident to say that is haplotype and should be removed to the haplotype TPF. Okay, there's another one we can place. Again, I'd be looking in the TPF to see if there's a gap here to accept this piece. If there isn't a gap, it could be a piece of haplotype, so we should check our coverage. I'll just put it there for now. This bit looks a bit suspicious. I would, well, if we look at the association between this bit and this bit, it's strong. But this bit has no association between here or here. I'd expect in the TPF that there'd be a gap here and a gap here. Assuming that is the case, let's just try taking it out and see if there's a better location to move it to. Okay, so it wants to go, wants to go at the point where we weren't sure of our join earlier. So this now confirms that we've got the scaffolding correct in this chromosome. So I'm going to put snap mode on and drop that into place. And now that's confirmed the whole structure of that chromosome. So that uncertainty is now gone away completely. Let's see if we can place the remaining pieces of shrapnel. So this is likely now very repetitive, tiny scaffolds, which we may very well not be able to place. Okay, we can definitely place this one. And that's the biggest one that was left as well, so that's good. Again, we want to be looking at our telomere tracks just to make sure we've got this in the right orientation. And we want to check it's not haplotype. So we look at coverage. So it drops down. It is very likely to be haplotype. One check we could do is to see if there are telomeres on the end of scaffold 58 and on the end of scaffold 6. Looking at the coverage, I'd say this is most likely haplotypic duplication, but we could have a look to see if the sequence of this scaffold is contained within this scaffold. So probably I'd be expecting to move that to the haplotype TPF. And this one has got a clear placement. Again, we'd do our normal coverage check to see whether this looks like retained haplotypic duplication or not, and then we'd either move it to the haplotype TPF or we'd place it in a gap depending on what we decided. And we could, if we decided it wasn't haplotypic duplication and there was a gap, then we would probably be using high glass to get the orientation of that piece because it's very tiny and Pretext doesn't have sufficient resolution. Just check the integrity of each of our chromosomes. I mean, on the face of it, this all looks really good, but we just need to have a detailed look at each chromosome. So to do that, we want to zoom in at a variety of scales and just check the structure. So this looks really good. One thing to note as well is it's quite normal to see association between the telomeres. So the right-hand end and the left-hand end are likely to light each other up, but to a lower level than the parts of the chromosome that are actually adjacent to each other. So you can see here signal, but it's low signal, and that's entirely normal to see. So no problems so far, it's all looking good. Okay, so there's an issue here. You can see a low level of association between this part, and strong association at each end of this part with this point. So we can edit our map, so we can chop this bit out, and we can move it, and we can flip it. And so I'm just having a very quick look now, otherwise I'll be here all day, but in reality, I would be taking a very detailed look at each chromosome. Okay, I'm happy with that. I think that is looking pretty good. And there we have it, one curated genome.